I'm going to give actually an introduction about brain parcellation. So I will try to give you an overview, which is actually a quite challenging task because brain parcellation, these are two terms that refers to a very wide range of methods to define brain region and network. I would say that we can consider that it basically finds its root in the cartography work. So we can consider that it has started with the pioneer cartographer like Brodmann, who try basically to identify brain areas by looking at local properties of the brain tissue. So for example, they look at cytoarchitecture under the microscope in order to look when there was a change in the pattern of cytoarchitecture as indicative of a change between two different brain areas. And then they manually draw a map of the brain. I don't know, no, we don't see the mouse here. And this work has been progressively automated. So now there are algorithms that automatically look at the pattern of cytoarchitecture and try to identify border to say when there is a border, when the pattern change, and then the map is automatically computed. However, the biggest avenue of progress or change came with the development of neuroimaging scanner in brain parcellation. Because with brain scanner, we had the opportunity to acquire a whole brain image in a few seconds. It means that we can look not only at local properties of the tissues, but also of, we could look at connectivity properties, so how brain regions interact together. So basically we compute connectivity information from the voxel and the vertices, and we use automated methods to define pattern and build map. In the last year, there have been a lot of development in the computation of connectivity features. So now we can compute many different types of connectivity features, and accordingly, we can basically derive a broad range of brain map based on these different connectivity features. And using quite big computing resource, then we can compare those maps or integrate them together within a multimodal atlas. I think this is what Matthew gonna talk about today. Multimodal parcellation, and we can use those techniques either for trying to understand the organization of the brain, or more in an application perspective. If we want to find a good is difficult to define, but to find an appropriate representation of the brain data. So, for example, if you want to predict behavioral scores based on resting state functional connectivity data of your subject, you do not want to use the connectivity information between all voxels, because this is a huge amount of data and you do not need all those data. So you might want to compress, to summarize the information with a parcellation map and then just look at the connectivity between parcels rather than between voxels. Well, as I said, there are really a range of parcellation approaches, and one way to characterize them is first at the technical level, so the type of algorithm that we use. The first parcellation studies that were based on histology were used to employ border detection or boundary mapping approach. So the idea is that an algorithm look at the pattern, and when there is a change, for example, in cytoarchitecture, it suggests that there is a border there. Then when connectivity was used as a feature for brain parcellation, some studies start using clustering or factorization approach. So here the basic idea is that the algorithm look at the similarity pattern of the voxel or the vertices and try to get to extract a pattern, a representation from there. We can also classify brain parcellation approach based on the type of feature they look at, and a broad classification that we can propose is between parcellation approach that look at local properties of the tissue, so properties that are expressed locally, such as cytoarchitecture, but also myelin, and also some properties that we can measure with MRI, such as the activation of the voxel across a range of different behavioral tasks. And these local approaches, local modality, can be contrasted to global approaches which look at connectivity information. So the global approach is basically the connectivity-based parcellation approach. Importantly, there is no assignment between the technique used, local or global, and the feature used, local or global. So basically, you can use any of those features with any kind of technique. For example, some authors have used a border detection approach with resting state data. This is what you see on the left inferior corner. And some authors have used a clustering approach with local features. So what they did is that they look within the amygdala the range of behavioral paradigm across which the voxel were activated, and then they cluster the voxel according to this information. Now I will focus a bit more on connectivity-based parcellation, because this is an approach that is so widely used, and most of the next talk will also focus on this approach. So the basic conceptual idea is that using neuroimaging scanner, we get information about connectivity of the voxel or the vertices, and we can study specific brain region or the whole brain directly. For example, if we are interested in the human hippocampus, we look at the voxel within the hippocampus, and if we look, for example, at the connectivity profile of these two voxels, for example, with resting state functional connectivity, we can see that they interact with different brain regions, so they have a different profile. Then if we do that for all voxels within the hippocampus, we can then identify a pattern in which the anterior voxel show a slightly different pattern than the posterior voxels. But there are many different types of connectivity measures that can be used, especially with the new development in the field. But the most frequently used approaches are the one illustrated here. So on the extreme right side, we have structural connectivity, which is estimated from diffusion MRI, so I mean anatomical connectivity, so probing white matter track. But we can also look at functional connectivity, so in terms of statistical dependencies between in the brain signal between brain regions. The most popular approach is resting state functional connectivity. However, we can also look at functional connectivity during active tasks, so when there is a behavioral constraint. And for doing that, we either need a group of subjects that would have been scanned across a range of different tasks. Why do we need a range of different tasks? Is because we do not want to have a profile of connectivity which is specific to a really specific behavioral paradigm. So we want a robust profile of connectivity across tasks, so we need for that a range of tasks. So either we have a group of subjects that have been scanned across a range of tasks, or we can use a meta-analytic approach. So basically we look at the activation data reported across the literature, and then we perform meta-analytic connectivity modeling. So this is what is illustrated on the left of the slide. So in order to get a robust pattern of co-activation of the voxel. So both resting state and meta-analytic connectivity modeling probe functional connectivity. They have advantages and disadvantages. For example, resting state functional connectivity is known to be highly susceptible to noise because we just look at fluctuation at rest under no constraint. But Ruby gonna talk more this afternoon about resting state functional connectivity. And finally, an approach which is a bit hybrid, I would say, is an approach that capitalizes on coplasticity. So the basic idea is that if we take a group of subjects and we measure, for example, their gray matter volume, and we look our gray matter volume between region covaries across this group of subjects. So when one region, the volume of one region goes up, the volume of the other region goes up as well. And this is an indication of coplasticity, and this can be used also for studying brain organization, for using brain parcellation, basically. So this is called structural covariance, but please do not confuse it with structural connectivity. Structural connectivity is based on wide matter estimation of wide matter track, and structural covariance is based on gray matter data, basically. So technically, how do we perform connectivity-based parcellation if we use a clustering factorization approach? First, we start with a region of interest, and we compute the connectivity between the voxel within our region, which are called seed voxel, and the voxel, all of the voxel in the brain, which are called target voxel. And this information is summarized in the connectivity matrix that you see here. And this is what we're going to use as an input for the clustering or the factorization. And basically what the clustering algorithm will do is that it will look into the similarity between the seed voxel, so between the voxel within our region of interest in their connectivity profile, which one have similar connectivity profile, how they are different from others. And then we will enforce a representation, a clustering, a subdivision into groups of voxel. And this is what you see here. We have to decide the number of clusters. So here we have defined 5 clusters, and you see on the upper corner on the left, when it is remapped to the brain, how it looks like. We can do that for different type of connectivity information. So you see on the top that what we get when we use structural connectivity, so based on diffusion data. And below you can see the pattern that we get when we use resting state functional connectivity and coactivation modeling. We can see that we have a very similar topographical pattern with an organization along 2 axes, a rostrocaudal axis and a ventrodorsal axis. So here the different connectivity measures converge quite nicely toward a similar topographical pattern. If you would like to use connectivity-based parcellation, but you don't want to start scripting everything and implementing everything, we have developed quite user-friendly pipeline in which, so let's say you have either diffusion data or resting state data in a sample. What you need is a target seed mask, so basically mask of your region of interest. If you have a specific region of interest, you can also input a target mask, but you can also use the default one. Then we propose different type of pre-processing of the seed and the target mask, but you can also just keep everything as default. And then the pipeline computes connectivity based on the most common tool with the FSL pipeline for diffusion, so PropTrax X2. And for resting state connectivity, we propose different type of denoising, because as I said, denoising is a crucial issue in resting state functional connectivity. Then we have a clustering based on the popular approach K-means. And then we have a range of validity metrics that are provided. So what those validity metrics give you as information is how well the clustering solution that you have, so for example, if you have a 2-cluster solution, a 3-cluster solution, a 4-cluster solution, how well this cluster solution represent your data, how well they summarize the information which is in your data. So they give you information about the quality of the clustering from a technical point of view, but they do not give you any information about whether this is biologically the true organization of the brain. So this is something to keep in mind. And you have everything in a report, and you have the nifty file of the parcellation solution you go for. If you want more information, please visit Niels Reuter Poster, who has done this work. He would be really happy to answer your question and explain further all the pipeline work. Because now I would like to address a more conceptual point, which is the difference between local microstructure and connectivity markers in parcellation. So as I said before, when we want to study brain organization, we can look directly at the properties of the tissues, the structural properties, with techniques such as cytoarchitecture, but we can also look at markers that probe large-scale functional integration and typically functional connectivity markers. If we do that, and we can do that, for example, in a brain region which is frequently studied, which is the human hippocampus, we know from a long time now that the cytoarchitecture of the hippocampus show an organizational pattern mostly along the medial lateral axis. So different subfields can be distinguished in the hippocampus, and they are arranged more or less along what I would call a medial lateral axis. Now, if we look at the organization of the hippocampus based on a connectivity-based parcellation approach, what we see if we use coactivation, for example, when subdividing into 2 and 3 clusters, so the basic level of subdivision, we see that the algorithm mainly distinguishes the voxel along the longitudinal axis. So we have a differentiation between anterior and posterior region. If we use resting state functional connectivity, we have something very similar, but we can see that it is really different than what we get with the basic model proposed by cytoarchitecture studies. However, so to summarize, to show it differently, so this is transparency of the brain and the hippocampus view, seen from the top. So based on local microstructure, what we see is this type of pattern with an organization more or less along the medial lateral axis. And if we look at functional connectivity, the primary organization that we see is along the longitudinal axis with an anterior-posterior differentiation. However, if we look at another connectivity modality, which is well less understood, I would say, which is structural covariance, what we see is also a differentiation between anterior and posterior region when we split in 2 clusters. But then when we split in 3 clusters, we can also see a medial lateral differentiation in the hippocampus body and tail. And this actually very resemble, oops, sorry, how can I go back? Yeah. This very resemble the differentiation between subiculum and CA subfield that we see in cytoarchitecture. So I hope this can convince you that different mapping features can reveal really different, can give completely different, not completely, but slightly different, at least, parcellation maps. So they are both convergence and divergence between the map derived from different features. So, to sum up, brain parcellation refers to a very wide set of methods to identify brain region and or network. It ranges from histology to MRI-based connectivity. It can be used either to understand or to represent brain organization and data. There are both convergence and divergence between mapping features. And if you want to easily compute connectivity-based parcellation, you can use CBP tool as a user-friendly pipeline, but it is also very flexible if you want to implement some changes. And this can allow you, for example, to look at the replicability if you have different sample or different data set, because this is also an important issue, and to further investigate the question of the convergence and the divergence between different connectivity features. Before finishing, I just would like to mention that I'm really happy to be in this symposium with great men and women speakers. And I'm really happy that OHBM promotes gender equality to further go in that direction, because it's not always easy for organizers to find women speakers. We know it, women are not visible. This is why we have created the repository for women in neuroscience. So basically, if you are a conference organizer, you want to organize a workshop, educational course, whatever, you go there. We have more than 1,000 profiles of women, and if you are a woman in neuroscience, please register there. And whoever you are, this is really important. If you hear talk that are nice, if you know a woman that you have worked with that you would like to give positive feedback, please go there and just click on recommendation and submit recommendation. This is really very important, and anybody can do it. And I know that there are a lot of women giving talk here at this OHBM meeting that have a profile there. So, and some of these sessions, such as Ruby, for example. So with that, I would like to thank my close collaborator, my research group in the Jülich Research Center, as well as my collaborator in Düsseldorf and beyond. And thank you very much for your attention.